Telomeres are the extreme part of chromosomes and one of the most fascinating topics of current science. It is believed that they could give great answers about complex and enigmatic processes such as aging, cancer and death. Telomeres were discovered in the 1930s by Hermann Joseph Muller, in the company of Barbara McClintock. This earned them a Nobel Prize. In 2009, researchers Elizabeth H. Blackburn, Carol W. Greider and Jack W. Shostak received another Nobel Prize in medicine for their advances in the same subject. It was previously believed that cells could divide infinitely. Then it was found that in reality, the cells could only divide a certain number of times and then died. The length of telomeres determines how close or far the cells are to die. What are telomeres? As we noted, telomeres are the extreme part of chromosomes. The word comes from a Greek root that literally means final part. It's something like the plastic part that is at the end of the shoelaces. They are considered the protective shields of the DNA of our cells. This means that thanks to them, to a large extent, the chromosomes keep their structure stable. That stability allows cells to divide the way they should. Telomeres play an important role in other functions. For example, thanks to them the cells do not adhere to each other. And this is very important for life itself. Among other aspects, telomeres also allow us to establish how much an organism has aged. Telomeres are the final part of chromosomes. Understanding telomeres, the cells divide constantly to regenerate the tissues and organs of our body. Each of these divisions causes the telomeres to be reduced a little, which are gradually becoming shorter. There is a point where they are so small that they can no longer fulfill their function of protecting DNA. When that point is reached, it means that the cells reached a state of old age and stopped dividing. The length of telomeres is measured in a unit called kilobase. At birth they have an average length of 11 kilobases. Upon reaching old age the figure is reduced to 4. The shortening of telomeres, and therefore their inability to protect DNA, seems to be the cause of aging and the appearance of age-related diseases. Between them, cardiovascular problems, neurodegenerative diseases, infertility, diabetes, some types of cancer, telomerase and youth, it is inevitable that telomeres shorten with each new cell division. However, this process can be moderated thanks to an enzyme called telomerase. Telomerase is capable of regenerating, to some extent, the length of these chromosome components. Stem cells are a kind of guardians of virtually all telomerase. However, the production of this regenerating enzyme decreases with age, and that increases the rate of cell deterioration. In theory, if telomerase production were maintained, we would have eternal youth. There is only one problem. There are some cells that have very long telomeres and produce a large amount of telomerase, cancer cells. These do not age and divide profusely. The conclusion then is that low production of telomerase increases aging, but high production increases the risk of cancer. So, in this respect, one of the problems of science is to maintain the generation of telomerase without causing cancer. There are already experiments with mice that have given encouraging results. It is not ruled out that the objective of conveniently modulating telomerase is achieved in the coming decades. The influence of habits. Various studies have proven that habits also affect the shortening of telomeres. As you can guess, the most harmful factors are consumption of sugary drinks, smoking, sedentary, lack of adequate rest, pollution. Likewise, it has been detected that chronic stress shortens telomeres, and that cortisol, the stress hormone, reduces telomerase production. Regarding food, there is no conclusive data, but a relationship has been found between longer telomeres and the consumption of vegetables, fruits, omega-3 and coffee. It has also been observed that babies fed exclusively with breast milk in the first 4 to 6 weeks of life have longer telomeres at 4 years of age. On the other hand, shorter telomeres appear to be associated with emotional depression.